Okay, thank you very much for the organisers for inviting me. I invited myself. I'm not thanking anybody. Okay, so I'm going to give you a bit of a whistle-stop tour because I thought, well, you know, this has been a fantastic opportunity for our lab. We've been, I would say, banging on about pseudotypes for about 15 years to actually be able to, you know, put our money where our mouth is and actually use these in the middle of an unfolding pandemic. So I thought I'd title this Pseudotype of the Pandemic, Vignettes from the Front Line. It sounds a little bit preposterous, but actually it's just a flavour in 1 or 2 slides per utility, should we say, of all of the things, well, not all of them, just some of the things that we've done during these last sort of 2 years. So I'm going to start with a little bit of history. So we were involved when I was still at UCL with SARS-1 and pseudotyping, and actually it was quite interesting. I don't know if Danny's still here, it looks like he's gone, but this is a snapshot from one of his first, I think the second BSI podcast he did early in 2020. You could see his face there, and he actually spoke in 1 slide about 2 of our papers, and I was alerted to this, and I thought, wow, this is really good. You know, these are papers from a long time ago, and they're being brought back, and suddenly the citations, you know, escalated once Danny sort of highlighted your paper. But essentially, they were showing what he thought, you know, he wanted to convey at the time was good news in that, you know, the antibody response was maintained over several months, and was reason for hope that if we were to get vaccines, that we would be able to boost that significantly. And then we've done bits and pieces with coronaviruses over the years. So in 2019, I did a study with the Jenner, which was looking at MERS vaccines, and particularly in the camel. And then also with NIBSC or MHRA, NIBSC with Jada and Emma, we've worked on the MERS antibody standard with the WHO. So we have done, you know, we've done quite a few things up until the point where SARS-CoV-2 began. So the remit of our lab is mostly pseudotyping, and from my side, it's mostly pseudotyping respiratory pathogens, respiratory RNA pathogens. And so on the top, I'm not going to dwell on this too much because Kelly, my postdoc, is going to talk about the flu story. But this top sort of figure summarizes about 10, 15 years of work in that this shows the full toolkit that you need to pseudotype all flu subtypes. And that's all A subtypes, but it's also influenza B and C and D, which we've now managed to do. But essentially, it shows you in the top left that you've got these basic set of plasmids, which are your spike or surface glycoprotein expression plasmid. You've got a core, which in the majority of the studies that I've been involved with is a lentiviral core. And then you've got a retroviral vector, which encompasses a reporter, luciferase, or GFP. With flu, you need some extra plasmids. You need a neuraminidase. For many flu strains, you need to cleave the HA, so you need to co-express a protease. And for some rare H7s, you also need to put on the M2 ion channel. And I've highlighted T2 here because T2 is TMPRSS2, and that in the production of flu pseudotypes is needed in the producer cell line. But if you now flip to the bottom, this is the SARS-CoV-2 and coronavirus story. Well, not all coronaviruses, those coronaviruses that do use T2 in the entry mechanism. And of course, in that case, the T2 has to be present on the target cell line, not in the producer cell line. So it's kind of flipped the other way. But essentially, the technology is very, very similar. So what did we do early pandemic? So my first big study I did early pandemic was actually here in Oxford. Believe it or not, it was a study commissioned by Peter Simmons, and it involved investigator Craig Thompson, who's actually going to join us for dinner this evening. We can't make the meeting. And they had this idea that they were going to try and get one of the first seroprevalence figures out there. I think we were amongst the first, with this crazy idea that they were going to use blood donor samples from Scottish blood donor service, and then these samples were going to bridge that period pre-incursion all the way through to the first cases, and we're going to do it using pseudotypes. But of course, none of these pseudotypes, we'd only really just got our assays going. And I remember the first day I arrived in total horror. A lot of pseudotypers had been called to bring in their batches of pseudotypes. So I had Dan Daly, a load of people who'd made like everything from 1 mil to 50 mils. And we got there, and Craig was sitting there with a big plastic bucket on top of a mixing thing, and every person gave him the virus, and he just literally poured it in. He said, well, we can't have all these different titers floating around, different cores and everything, just chuck it all in, and then we'll estimate the titer and hope for the best. And we tested 1,000 sera over 3 days. We had the free reign of the Medawar. We used 3 floors, 3 different hoods, different people did different jobs, PhD students were running up and down, taking things. It was very challenging, but it was very, very exciting. And actually, the idea was that we do the work, and then on the last day, give all the data to Peter Simmons, he'd write it up and answer, and we'd be out in a week. That didn't quite happen. But we did get a few positives, and we estimated the seroprevalence rate of about 0.6. And again, this was also featured on Danny's podcast. So we were quite lucky. So he featured the Peter Simmons study, which is the one on the right. There was another study from the lab that came up, 3% seroprevalence rate. We got a 0.6, and there was another American study later on with about 7%. And actually, because of discussions over publications and authorship and everything, you can imagine with this study, the study ended up being tweeted as a publication. So the actual tweet is the original publication. And this is the publication here. It's a map of Scotland, where the different samples are. It's got everything you need of a paper. It's got methods, data, and it's at the bottom preliminary data of the Oxford Immunology Consortium, Scottish University of Kent, and others. A full author list to follow on submission. Now that paper actually took quite a long time to come out. And the paper that actually came out is in neurosurveillance, which was a detection of neutralizing antibodies to determine population exposure in Scottish blood disease. And so that, again, that was one of the first determinations. And then we thought, okay, well, we really need to get involved in putting out some good quality methodological publications now for this assay. And that actually brought both myself and my colleague Simon into the lab. Here's evidence that we actually did some experiment. We had a nice consortium with, we used some PhD students on our campus that were working in plant sciences. This is also in collaboration with Emma and with Jada at LIBSC, and also with Craig at Oxford. And we put together quite a comprehensive set of protocols for SARS-CoV-2, as did many other labs down the line. The next thing we did is we got involved again with Jada and our former boss, Mark Page, to be one of the constituent labs that fed into the international standard. And you can see on the picture on the top, this is with myself, with Mark Page, with the standard. And an old picture to embarrass Ed was when we first got into this standards game. I can't remember what year. We went to a meeting for Ebola standard, didn't we?
It was like yesterday night.
Yeah, in Geneva. And the meeting they actually held in a hotel. And we got to the end of the day and it was a dinner, and Ed and I said, we're really disappointed. We never got to see the WHO. We've come all this way. And some people said to us, well, look outside, it's chucking with rain. We said, we don't care, we're going to the WHO. So we trekked all this way in the pouring rain and actually got a photo outside the WHO. And so that was the first time we sort of got involved in this. And then, like many other people, we also got quite heavily involved in what I call variant chasing. So we had quite a nice cohort of samples via our we had a collaboration with the Royal Papworth via a Cambridge-led HIT consortium, and they had a very nice cohort of healthcare workers that had been vaccinated and followed. And then, of course, their sera was tested against all the different variants that were coming out. And like many other groups, we showed that, you know, as you progress through the variants, and this is split here into the different types of vaccines, you generally got a lower and lower response as you progress through alpha, delta, and so on and so forth. But it's the right-hand side figure that's really the more important. There's lots of studies now that back this up, that people who had prior infection before they were vaccinated make a much higher neutralizing antibody response in those that don't have prior infection. So then on to correlative immunity. So, everyone was asking, okay, there's all these assays out there, but what's the cutoff at which you would consider yourself protected using assay X or using assay Y? And so as part of the HIT consortium in Cambridge, we were down initially to work with siren samples as well. So this is the UK siren study, so it's a reinfection study, and it's looking, at unvaccinated individuals and, at what level of neutralizing antibodies are you then protected from subsequent reinfection. And for me, it was a really important study, even though we didn't lead this. It was very important because the two main assays used were live virus neutralization and pseudotype virus neutralization. And it comes up with a cutoff. And so the cutoff was 1 in 40 for the live virus and 1 in 100 for the pseudotype. And actually looking back at all of our data, 1 in 100 was what we'd arbitrarily set our own cutoff. So actually it was quite lucky because it was about right. And other studies have come quite close. So of course now there are a lot more studies that actually put this into international units and take this a lot further. But it was a nice study to actually show that this could be used for correlative immunity. And then it really got us down the line of how do these assays compare. And this is some data from that siren paper, so this is not our data. Just to take you through very, very quickly, they compared on the top right pseudotype neutralization versus a Rog-S, so it's just an ELISA-based assay, and then in the middle, live virus neutralization, but more importantly to us, some nice data with a decent sample size looking at the correlation between live virus neutralization and pseudotype virus neutralization. So then I think Miles is responsible for this, that you commissioned a special issue in Frontiers on this very issue of correlating assays. I think in conjunction with Sept and a few others. And I had a postdoc, Diego Cantoni, who's now at CVR. And we kept getting asked this question about how things correlate. And we said, one day what we should do, we should go through the literature and download every single paper for every single virus where there is a correlation between live virus and pseudotype virus, and actually do a meta-analysis. I said, well, I have no idea how to do a meta-analysis. Go and do it. And then when he left, joined Glasgow, and in the evenings, he's bored, he needs stuff to do. So he's actually done this study, and even though he's working in a different environment, and it should be out very, very soon. It's been accepted. But essentially on the left, you can see the different types of assays. So, with live viruses, obviously you've got the containment issue. Many of the viruses contain level 2, 3, or even 4, like Ebola. On the right-hand side of that figure, you've got the pseudotyping systems, and the main ones are obviously the Lenti and the VSV core. And then these are usually evaluated either with a luciferase or with GFP, either by FACS or by imaging or by luminescence. And on the right, you can see, you don't need to really look at these numbers, but this is just listing a huge amount of papers that we found, with the N being the number of samples that were tested. And then on the right, looking at all the different correlations that people got. And in total, it involves about 1,238 data points by both pseudotype and live virus neutralization. And we calculated from that pooled correlation of 0.86, which I think is actually good. Then don't ask me about any of these assays because I won't be able to tell you, but we did a forest plot which apparently tells you whether there's any kind of publication bias. And so all of the data points need to be in the top of the funnel, which they are. And the other assay, which 4 of the papers used, was a Bland-Altman assay, which applies to determine agreement between 2 metals that are measuring the same thing, so in this case, neutralization. And 4 papers for which there was a Bland-Altman analysis in, all had very good correlation between both assays. And in fact, I'd previously done a Bland-Altman assay with Vismaderi colleagues who are also in the audience, with Emmanuel Montemoli in Siena, where they actually had the live virus set up pretty early on, in early 2021, and we did a nice study comparing the pseudotype assay with the live virus assay, with one of those analyses. And the last bit of this correlation paper is quite nice, because what they actually did is split the correlations between the different types of cause. So you've got HIV versus VSV cause. Obviously, there's different numbers of studies, but you can see the Pearson's correlation using the different cause, and then also splitting it out also into reporters. So enhanced GFP, GFP, luciferase, PRNT, and CAP, and then look at them like in that way. Obviously, you've got to be very mindful that for some of these, there's very few studies that use a particular reporter, so luciferase is by far the most popular. But then really most interesting is sample type. When you're looking at different samples, like whether you're using plasma, whether you're using sera, and seeing how the correlations pan out across those as well. So I think there's a lot more to be done. A lot of papers we had to exclude, which they didn't have a direct correlation within that paper, and also anything that wasn't strictly what we defined as pseudotype we excluded. So there's some other types of recombinant assays, of these surrogate neutralization assays, like the GenScript C-class assay that we had to discard because it's not it doesn't fall into one of the two categories. So I think somebody else could follow on this with a much more comprehensive all neutralization assays. We only focused on two types of neutralization assays. And just to kind of round off, just to look at a few other studies that we got involved with. Obviously, we're all sat here, without masks now, but there are a lot of people that are still shielding. And they're mostly immunocompromised people who have various different deficiencies. And this is just a series of studies that we did, again, with a cohort from Papworth Hospital with severely immunocompromised individuals. And you just can see very, very clearly when you look at this graph at the top, where you've got the immunodeficient outpatients, and look at the scales on the top, there are some differences. And then healthcare workers with no prior infection, healthcare workers with prior infection. You look at those huge titles that we have and take for granted compared to the immunodeficient patients. And then we did a follow-up study, which is even more striking. And this is just the final data from that. On the left, you've got healthcare workers. So PV2 is vaccination 2, PV3 is vaccination 3, so that'll be first booster. And then the fourth bit is PV3 plus infection, so it's prior infection, then boost. On the right, we look at the immunodeficient patients. So I'll just read you this. On the left-hand side, only 3% of healthcare workers failed to neutralize the vaccine ancestral strain, but 39% failed to neutralize BA.4/5 after the second vaccine. But then only 13% didn't do that after the third vaccine or the first dose. So that's pretty good news, considering that vaccine's a Wuhan vaccine. You're even getting protection from hospitalization against the Omicrons. But then you look in red, this is in the immunocompromised. After the second vaccine dose, 43% of the immunocompromised cohort failed to respond to the ancestral strain, and 83% to BA.4/5 respectively. And after a third immunization, of the people involved with follow-up, 70% still failed to mount an immune response capable of neutralizing BA.4/5. So that's quite shocking. And then a third study that we actually did again with the same group was this time not just looking at neutralizing antibody, but also looking at T-cell immunity as well. And there the picture is a little bit more hopeful, in that often that lack of neutralizing capability in the immunocompromised would made up for T-cell response. But again, it boils down to which vaccine you have. It's actually better to have channels vaccine for T-cell responses in the immunocompromised rather than the mRNA. Again, it's only one study. And then I've been involved also, which I've heard famously called Gavin Screeton, it's not in the audience, but I've heard his series of papers called the Screeton cell line, which basically means you have loads of amazing monoclonals, and when every variant comes out, tests the hell out of them, and then you publish it in Cell, hence the cell line. And we were involved in a lot of this with a lot of these different assays, but I just show this particular study because again, it really shows this, as Dan Norton spoke about earlier with Omicron, or Omicron. When you look at, so on the top, above the line are Gavin's panel of monoclonals. You look at the bottom, these were the licensed, commercially available monoclonals. So you look at that first column, Victoria, beautiful neutralization by, the Oxford cohort, the Oxford collection of monoclonals, but also by all the commercial ones. Then the next line is BA1, so the first Omicron. then BA1.1, BA2, BA3, you can see a total lack of neutralization. So all of these companies that invest all this money in all these lovely monoclonals, they don't neutralize past Victoria. And this is a real problem. It's an ongoing problem, but you've still got a lot of groups working on it that are, but you can't just make a monoclonal and hope that it's going to last for a long time. I think Danny alluded to that, would say that some of these monoclonals may in fact be useful for treating certain people with long COVID, which is good and would be a good use. But it really depends which, what was the virus that infected you. And the one thing that we've done towards the future, sort of pandemic preparedness, is looking at new spillovers and how we can use pseudotypes to try and look into the future. So if you can identify by whatever means a particular coronavirus that you think may spill over in the future, we can take the spike of that, we can make a pseudotype, and then we can test current sera from vaccinated individuals and ask the question, if that were to spill over, would our current vaccines protect? And actually, we did this with one of the bat coronaviruses, RATG13, and we actually cloned with this is a collaboration with Dan and Bailey at Purbright, and they actually cloned the spike, made some pseudotypes, and then we tested a range of healthcare worker sera pre and post vaccination, and with prior, and also with prior infection. And we also looked at the NIBSC panel, and we found to our surprise that actually all the sera that we had from wherever we took it, and all the monoclonals, and also the reference sera, all neutralized the RATTG pseudotype better than SARS-CoV-2. So actually, it was the RATTG13 that pseudotype. We would actually be lucky, but that might not always be the case with other coronas. But I think quite a lot of labs are now taking this forward on a huge scale, and they're basically looking in bats, looking at all the coronavirus types that may spill over, making something like pseudotype libraries, and then screening huge amounts of vaccine sera to say, how well would we be protected, or do we need vaccines that concentrate more on this clade or other clade? And of course, that's where the pan-Sarbeco vaccines will come in. And then just one final thing, which has only just come out last week, that I actually did in conjunction with the University of Cardiff Medical School, Alan Parker, who actually works on gene therapy most of the time, and also a company that makes these electrostatic ones. And the whole idea is that during surgery, there's a lot of aerosols come out of different wounds, and these can carry huge doses of virus, putting surgeons in increased danger during pandemics. So what they developed, this company, was essentially one system. So you've got on the left your viruses in droplets, so aerosolized viruses, and then using this one, these aerosolized viruses become negatively charged, and then you've got a positively charged wand which captures these and inactivates them. So what they did is they built this setup in the lab. I've seen it; it's quite strange. They've got this big kettle. They put viruses in there. We weren't allowed to use real viruses. We did adeno from the gene therapy side because they had the adenos, and that was an example of a non-enveloped. And then we used SARS-CoV-2 pseudotypes and aerolite, and then see if we could get them to attach to the wand and then be inactivated. And surprise, surprise, it actually works really well. So you can see on the top here, you've got the DNA, which is the genomic reduction. And on the right, you can see the actual transduction reductions, but you can see it much better visually. If you look at the bottom right, so you can see the non-exposed is just the virus only with the GFP. And then you have your wand, which is off. You can see a huge amount of green GFP cells. And when you put the wand on, you reduce that. There's about 99% reduction. And the summary is that the bioaerosols released from patients can obviously facilitate virus spread, but you can use a simple technique like electrostatic precipitation to capture and then inactivate viral protein particles. And this could be a viable means to reduce nosocomial infections in pandemic situations. So I think overall, I hope I've given you a flavour that pseudotypes were once a very niche area, but actually they've been used by our lab and others for assay development, for seroprevalence studies, for looking at antibody standards, testing vaccines and variants, looking at risk populations, contribute towards correlates of protection, studying NABs, preparedness, and also now maybe have utility towards testing different devices that can be used in the hospital settings to minimise risk. I'd like to thank all the members of my pandemic team that was last year. Very fortunate to have a very wide international consortium of collaborators. So we've got postdocs from Brazil, Portugal, the Philippines, Italy, and then on the right, this is post-pandemic when we'll get together without the horrible masks and actually go for a meal in London together. So this is mostly work presented from my lab, also with our biggest collaborators, which are Cambridge, with the University of Glasgow, where Diego is. We've had various generous pots of money from various funders that are funding us for both the influenza side and also the SARS-CoV-2 side that they've kind of been running in parallel. So thank you very much.